Hello friends, welcome to Cultura channel. I am Varsha Gaitonde and in this video we shall discuss the history of DNA. The history of DNA starts with Fred Mischer's work in 1869, who worked with pus cells, that is nucleic acid, and named it as nuclein. He could not name it as nucleic acid. Then in 1889, Altman, who demonstrated nuclein was acidic and named it as nucleic acid. So the exact meaning for nucleic acid was given by Altman in 1889. In 1928, Griffith made and found the transformation principle with Streptococcus pneumoniae. Subsequently in 1944, Griffith, Avery and these people contributed for transformation principle. Okay. So in 1944, Avery, McLeod, McCarthy, they also worked with Streptococcus pneumoniae, used different enzymes like DNase, RNase and protease. In 1952, Hershey and Martha Chase, they made blender experiment, used phases worked with T2 phase. They postulated that if one could separately tag both the DNA and protein in the phases, then one could follow the DNA and protein through the phases replication process. They followed three processes. One is blending, then agitation and then centrifugation. Using viral particles with the DNA triggered labeled with P32 phosphorus and protein labeled with S35, they showed that only DNA of the virus entered the cell in significant amount. That means DNA is somewhere. A transformation principle was perfectly reported by Hershey, but the preliminary work was done as DNA as the genetic material by Avery, McLeod and McCarthy. This result supported a role of DNA as a genetic material and refused the role of protein because S35 failed to pass to the next progeny or next generation. It just remained in the ghost cells. If we talk about the discovery of DNA, as you know, in 1953, Watson and Crick contributed for this. For Watson and Crick's double helix model, four scientists mainly contributed. The first was Rosalind Franklin, who is called as Dark Lady of Genetics, worked with X-ray diffraction pattern which was obtained by Watson and Crick by her postgraduate student Raymond Gosling. The second scientist is Wilkins, who proposed helical corkscrew-like shape of DNA based on X-ray diffraction pattern along with Franklin. Next is Alexander Todd, who determined that backbone of DNA molecule contained repeated phosphate and deoxyribose sugar goof. Then Chargaff, who found the amount of DNA and its four bases as adenine, guanine, thymine and cytosine. Remember, these four scientists are responsible for the Watson and Crick's double helix model. Okay. By their findings in 1953, April 25th, they made complementary anti-parallel double helix model of DNA, which was published in Nature. In 1962, they awarded with Nobel Prize. In 1953, May 30th, these two scientists developed their idea about genetic replications in their second article published in Nature. Remember, in their first publication what they made, they told about the double helix model of DNA, and this was published in April 25th, 1953, and the second paper was just 1 month after that, May 30th, 1953, that is about genetic replications. In 1964, Holliday, who gave hybrid DNA model of crossing over. In 65, Whitehouse, who gave Polaron hybrid DNA model of crossing over. That means they concluded on the basis of single stranded break and double stranded break. Odile was Crick's wife, who drew double helix structure. She is also very important because she was the inspiration to get this double helix structure. In 1967, Gollert, Lehman and Richardson, they gave the very important enzyme DNA ligase. In 1968, Werner Erber developed a restriction enzyme. Erber hypothesized that bacterial cells produce two types of enzymes. One is called as restriction enzyme that can identify and cut foreign DNA. Ligase is a molecular glue which seals the nicks and joins together the cut ends, but restriction enzyme which gives internal and external cuts. Okay, so there are two types. One, that is restriction enzyme that can identify cut foreign DNA, and make modification enzyme that recognizes the host DNA and protects it from cleavage. This hypothesis was proven in an experiment in which enzymes were isolated from E. coli. The modification enzyme methylase protected DNA of the bacterium, while the restriction enzyme chopped off phase of the DNA. In 1970, Hamilton Smith, who gave purification of type 2 restriction enzyme. He worked with bacterium Haemophilus influenzae in 1970s. In 1972, he successfully purified the first site-specific type 2 restriction enzyme, that was HIND2, from this bacterium. He and his team also identified 6 BP phase DNA sequence that HIND2 recognized for the site-specific cleavage. This understanding of how restriction enzyme cuts the DNA and how host DNA works to protect itself is the basis for modern genetic engineering as well as for genome editing, especially for the CRISPR technology. In 1971, Paul Berg, did the gene splicing experiment which paved way for recombinant DNA technology. He became the first scientist ever accomplish creating recombinant DNA from more than one species, which come to be known as cut and splice method. Remember, the Paul Berg method was cut and splice. He cut the DNA from two viruses, creating sticky ends. DNA was then incubated. The ends would anneal on their own, and addition of DNA ligase would seal it. This proved the validity of theory that it was possible for any two DNA molecules to be covalently joined together. This achievement was considered a fundamental step in the field of genetic engineering, which was the biggest stepping stone towards creating the recombinant DNA. Paul Berg awarded Nobel Prize in Chemistry in 1980, showed with Gilbert. In 1971, type 2 restriction enzyme was used for mapping DNA tested on SV40. For this enormous work in 1978, Warner, Nathans and Smith was awarded with the Nobel Prize. In 1972, Stanley Cohen and Herbert Boyer, they created recombinant DNA technology, which became basic for the genetic engineering. Then in 1975, George Kohler and Cesar Milstein, they contributed for hybridoma technology, which revolutionized the diagnostic work. For the production of monoclonal antibodies, this hybridoma technology is helpful. That means hybridoma technology is nothing but it is the process for getting the monoclonal antibodies mainly. In the same year, 1975, Edwin Southern gave Southern blotting technique, which is helpful to detect specific DNA from the DNA samples. Following in 1981, Thomas Wagner was the person who found the first transgenic animal, or who has given the first transgenic animal, rabbit, into mouse DNA by microinjection. That means he did the genetic engineering work. In 1983, Carey Mullis, who developed the PCR technology, which is popularly called as People's Choice reaction also, which is helpful in cloning the DNA to multiple numbers. For his work, he was awarded with Nobel in 1993. In 1984, Alec Jeffrey, contributed for DNA fingerprinting, and in India Lalji Singh is called as father of DNA fingerprinting, but in world Alec Jeffrey is called as father of DNA fingerprinting, and he contributed during 1984. In 1999, first human chromosome was sequenced, that is chromosome 22, and 2000 onwards, including modern organisms like Arabidopsis, rice, maize, model organism Arabidopsis, rice and maize. Till today, almost all field crops, commercial crops and relevant species are sequenced and resequenced to find out the fine rare alleles and genes in the genome. That means from DNA now the main concentration is on some other things like alleles as well as genes. To summarize, these three contributions are very important. First one is Griffith's contribution in 1928, that is transformation principle, which was the work on Streptococcus pneumoniae. Then the work of conjugation by Joshua Lederberg and Edward Tatum worked on bacteria, that is Streptomyces. Then the third work was on transduction in Salmonella by Zinder and Joshua Lederberg in 1952. Friends, I think this topic is helpful to you, and if you like this video, please do not forget to share, subscribe, comment and like. Thank you once again.